Last speaker is Dr. Atsuko Yamashita from University of Osaka. So the title of her talk is Versatile but Specific Chemical Recognition by a Model Taste Receptor Protein, medaka T1R2A, T1R3. Please start.
Okay, thank you for the introduction. And I'd like to thank the organizers and advisory board of this symposium and Sophion Bioscience for giving me an opportunity to talk in this exciting symposium. I moved to the University of Osaka 6 months ago, and Osaka is a town known for kuidaore. That means in Japanese old saying, people in Osaka love to eat so much that they ruin themselves with extravagant spending on food. Therefore, please forgive me for speaking about gas station today rather than ion channels. So to enjoy these foods, we should detect diverse chemicals in foods. To do that, we have taste receptors in our oral cavity categorized into 5 basic taste modalities such as sweet, umami, bitter, sour, and salty. These receptors are conserved in jawed vertebrates, and we recognize nutrients as sugars and amino acids, and toxic or decomposed substances as beta substances of proton, or electrolytes as sodium ion. So the functional requirement of taste receptors are they should recognize diverse chemicals by a limited varieties of receptors. Therefore, in many cases, taste receptors have broad substrate specificity. For example, we humans have only a single type of sweet receptor, that is T102, T103, but we recognize various different kinds of sugars, such as monosaccharides and disaccharides, or artificial sweeteners, such with completely different chemical structures with sugars or even proteins, and then induce the same consequence, that is the signal transduction to the cytosol of the taste cells. So here a question arises: how do taste receptors recognize diverse chemicals and induce signal transductions? So we are focusing on taste receptor type 1 protein, T1 or tasphanols, that serve as sweet and umami receptors in humans. And we are working in the field of structural biology. However, a bottleneck for structural analysis of T1 are sample preparation. Even for focusing about the soluble extracellular domain of this protein, most of genes cannot properly folded and properly expressed well in most of the expression system. So we employ the fact that these receptors are conserved among jawed vertebrates, and we screened and found that medaka, that this small fish, T102A, T103, only express using the insect cell expression and took proper folding state, and successfully solved the crystallographic structure 8 years ago. Therefore, the ligand binding domain of LBD of medaka T102A, T103 has served as a model T1 protein amenable to recombinant protein preparation and protein level analysis until very recently. This receptor is an amino acid receptor, so we first asked that how diverse are the chemicals that medaka T102A, T103 LBD recognize? To address this question, Takashi Yoshida employed this thermal shift assay that can be evaluate the ligand binding in terms of this thermal stabilization, that is the increase of TM temperature. And here is the result. He showed that medaka T102A, T103 LBD binds diverse L amino acids irrespective of their physical chemical properties. And because taste receptor does not exist not only in oral cavity, but actually they are also expressed at the extraoral organs inside the body. Therefore, Hikaru Ishida extended his question to the substrate of the medaka T102, T103 ABD for other bioactive compounds such as taste substances for humans as sugars and nucleotides and vitamins and neurotransmitters, hormones, or polyamines and aminoglycosides. And unfortunately, he didn't find any clear binding of these chemicals, especially even GABA, that is gamma amino acid, cannot show the clear binding to this medaka T1 proteins. However, if we try the metabolites, although most of them show clear binding, but only L-citrulline and probably L-ornithine for some extent show the binding signal to this protein. Therefore, medaka T103 ABD is found to be specific to L-alpha amino acids because these compounds are L-alpha amino acids. But however, with diverse substituent groups. So going back to the structure, we already reported that the ligand binding pocket of T102A share a large space, actually much larger space than the ligand amino acids. And here I show the electrostatic potential of the surface of the ligand binding pocket, and it shows the mosaic pattern, such as positively charged region shown in blue, negatively charged region shown in red, and neutralized region in white. So this structure characteristics explains why this protein can accommodate diverse substituent groups of L-alpha amino acids. Then we asked that how does medaka T102, T103 induce the same consequence that is the signal transmission to cytosol from diverse amino acid recognition? Because T1 class C GPCRs and other class C GPCRs, such as metabotropic glutamate receptors, are proceeded, the structure biology of them are proceeded to taste receptors. Here I show the result of mGLAU2 reported by Bailey Wu's group in China. And they reported that upon binding of the agonist at the ligand binding domain portion, this LBD protein showed the conformational change. And this change is transmitted to the transmembrane domain, allowing the G protein coupling at the cytosolic side. Actually, we already reported that this medaka T1 protein also showed the conformational change by using the fluorescent fusion protein of this protein and analyzed the FRET. And many amino acids can actually induce the increase of the FRET signals. And also we confirmed that these amino acids actually induce receptor responses when we analyze the Furex receptor. And we analyzed the crystallized glohic structure of the protein in the presence of various these 5 amino acids, and all of them, actually the alpha amino and alpha carboxy group of them take just the same position in the receptor, taking in the same configuration, and inducing the same conformation of the protein, just similar to the agonist bound state of mGLAU2. So next we asked that what is the key parts in L-alpha amino acids for recognition by medaka T102A, T103 ABD protein? So we first tested L-alanine and D-alanine binding, and we confirmed that this protein is L-enantiomeric. Then we tested the compounds lacking the carboxy group or alpha amino group of L-alanine, and none of them showed binding. So alpha amino and carboxy groups of L-amino acids are critical to be recognized by the receptor. Then we tested the modified amino acids, such as methylation at the amino group or methyl esterification at the carboxy group, and they show the binding for some extent. Then we asked the rigorousness of the recognition and by analyzing the affinity of these to these compounds, and found that the modification at the alpha carboxy group or the alpha amino group impairs the recognition, like tenfold increase of the KD values. However, but the impairment was not critical. However, if we tried 2-methylalanine, that is the substitution of alpha hydrogen with methyl group at the alpha carbon, the impairment was quite severe. That is the more than 100-fold increase of KD values. So when going back to the structure, the alpha amino and alpha carboxy group was recognized by hydrogen bonding networks serving by two serine residues, serine 142 and serine 165. And this alpha hydrogen is likely recognized by CH pi interaction just beneath the substrate phenylalanine 213. And considering about the fact that this alpha hydrogen substitution with methyl group gave severe impairment, these CH pi interactions is also important for L-alpha amino acid recognition. Importantly, these three residues are conserved among amino acid sensing ternals, including our human umami receptor. And these two serine groups is located at the upper subdomain of the ternal ABD protomer, that is LB1 portion, and this aromatic residue located at the lower portion of the subdomain, that is LB2 portion. Therefore, interaction between the common groups of alpha amino acids, such as alpha amino group, carboxy group, and alpha hydrogen, and the key residues in ternals are necessary and sufficient for conformational change of ternal LBD, irrespective of the chemical structures of substituent groups. Next, I'd like to go to another topic about another functional versatility in ternals found in structural analysis of this medaka protein. During the structural analysis, we found a chloride ion binds at an allosteric site to the amino acid binding site in ternal 3 subunit. And chloride is a component of sodium chloride, and sodium chloride inducing taste is known as salty taste, and that is induced by the interaction between sodium ion and salty receptor ENaC. However, an enigmatic phenomenon was reported like 60 years ago by Bartschak in United States by human sensory test. So we sense salty tastes around the concentration of 100 millimolar sodium chloride solution, like in the solution in miso soup. However, in the lower concentration, that is the lower than the detection threshold of ENaC, like 10 millimolar or less, people sense the sodium chloride solution as sweet. And this, nobody knows what underlies this mechanism. So, and this sweetness is inhibited by Gymnema sylvester, that is a human sweet receptor inhibitor. Because ternals serve as sweet receptor in human, so we decided to pursue the actions and physiological relevance of this chloride binding to ternal 3. First, we asked that what happens when chloride binds to ternal 2A, ternal 3 LBD. We again employed this FRET system and found that solely chloride addition actually induces FRET signals and in the concentration of around millimolar range. So chloride binding induces conformational change of ternal 2A, ternal 3 LBD, similarly to the amino acid that is canonical taste substances for ternals. And we extended the binding specificity of this site and found that not only chloride, but larger halogen ions such as bromide iodide also bind and can induce the conformational change of this protein. So we go back to the structure and found that this chloride ion is coordinated with amide group of the residues 146 to 149. and coordinated with hydroxyl group of threonine 105. And when Nanako Atsumi mutated this threonine to alanine, no more conformational change was observed, even application of chloride as well as amino acids. So the chloride binding site in T103 here is found to be responsible for the conformational change of T102A, T103 LBD. Importantly, this amino acid sequences is conserved among T103s in most of other animals, including human and mice. So we asked that whether chloride detected by the taste sensory system in living animals by employing the fact that the chloride binding site is likely conserved in mouse T103 too. Therefore, we decided to collaborate with Professor Yasumatsu at Tokyo Dental Junior College and analyze the single fiber recording from mouse taste nerve that is called the tympani nerves. So Professor Yasumatsu first identified sweet nerve that is connected with T1 nerve expressing taste cells by the fact that sucrose addition or amino acid addition can induce nerve responses, and the responses were inhibited by glurmalin application, a T1 nerve blocker. Then, when she applied chloride ion to this nerve, chloride addition can indeed induce receptor nerve responses, and the responses were inhibited by glurmalin application. So we confirmed that these nerve impulses were induced regardless of the counter cations in the concentration of range of the millimolar range, and all of these responses were inhibited by glurmalin application or was not observed in T103 knockout mice. So we verified that chloride-induced responses in mice called the tympani nerves via 10 Rs. We further asked whether mice preferably perceive chloride by taste sensation as our sweet or umami sensation mediated by T1 nerves. So we tested two-bottle test and found that mice prefer chloride-containing solution than normal water, and this preference was attenuated by glurmalin application. So we concluded that chloride is preferably perceived through taste signal transduction mediated by T1 nerves. Recently, a breakthrough study was reported from the Charles Zucker's lab in the United States. That is, they show that is the kind of really groundbreaking work in structure biology of T1 nerves. That is the first full-length structure of the human sweet taste receptor. Here, they reported that at the T1 nerve 2, an artificial sweetener, sucralose, that is a derivative of sucrose, bind at just the same site where we observe an amino acid binding at the medaka T102A, T103 LBD proteins. And moreover, although Zucker's group didn't mention anyway, or even not modeled, but when we inspected the density map deposited by Zucker's group to the database, we found that some additional blob density was observed just corresponding site. We observed the chloride binding at our medaka protein. Therefore, we are assuming that this is actually the chloride binding and explain the human sensory of this sweet sensation induced by sodium chloride. And further study should be necessary to address this binding results. So I'd like to summarize the study. So the T1 nerve proteins equips multiple chemical binding sites, which is L-alpha amino acid or other nutrient binding sites, and halogen ion binding site, which can accommodate a diverse range of chemicals. And importantly, these binding sites just locates in the vicinity or inside the portion where the pivotal region where the LBD show the conformational change, such as the inter-subdomain cleft or the intermolecular dimerization site, which is known to reorient about the agonist binding in mGlu2. So they have mechanisms which couple binding or release of chemicals with conformational change of the protein. So these structural characteristics may underlie diverse chemical recognition covering organic compounds such as nutrients that is considered about the canonical taste substances for T1s. Moreover, to ions, that is a new observation, but the electrolytes serving as electrolytes for us. So this study is done at my previous affiliation, Okayama University, and I'd like to thank all the collaborators of this work, and thank you very much for your attention.
Thank you very much for the very interesting data. So any question from the floor?
Thank you very much for the beautiful talk. It's pretty interesting. I have a question about the second topic, about the chloride binding to the T1R. So if the chloride binds to, so it activates the receptors in the nerves, right?
Yes.
With the moderate concentration, like 1 to 10 millimolar.
Yes.
So how about the high concentration with 100 millimolar? Still activates the sweet neuron?
Yes. So actually, this is my mice data. So we did not take the, but yeah, we take 10 millimolar or something, millimolar or something. So we took higher concentration. It's still activating the receptor.
Yeah. So for example, like a sodium chloride, we don't really taste the sweetness.
Right? I know. But probably this is the kind of, so we really sense in the brain, like, I don't know, higher signals is more precededly recognized. For example, we usually sense salty sensation as more intensely than this weak sweet sensation. Actually, this is quite weak sensation as a sweet taste because, for example, usually sweet taste sensation nerve impulses like, I don't know, 100 per 10 seconds or something. It's kind of weak or thin sensation as a sweetness. So probably this salty sensation kind of overlap to recognize by us. That's kind of speculation.
Thank you. I have one silly question. Have you ever tried the sweetness of the NMDG chloride solution by yourself?
By myself, yes, actually. So I'm really convinced about that. So when don't tell to our student, but I really tried, I myself tried to taste NMDG tasteless. I tried NMDG chloride, very intensely sweet, amazingly.
Good to know. Thank you very much.
Any other questions? So in can I question? So in your results, so NMDG chloride also solely induce response. So how up the NaCl is, if you apply the sodium solely, so without chloride, it induces the response, and if so, the response is higher or lower than NaCl?
So it's kind of, we are working on that. And it's, for thinking about, we have additional sodium, we actually, we have several experiments working on it. in medaka, using medaka T1R protein doesn't show any clear conformation change or on just upon addition of the sodium. However, we kind of observe the sodium application induces some nerve impulses. However, it kind of confused us that we didn't show not really clear addition of the sodium chloride application compared to the sodium chloride application.
I was wondering if the chloride can enhance the response of the sodium or just together in the same intensity of the response is induced, either sodium alone or sodium together with the chloride.
So yeah, I think that's kind of a good point, and we are working on that. And actually, this is a chloride binding site. It's allosteric to amino acid, but actually we also have sodium binding site here. Now we are working on. It's allosteric. So I think probably, I don't know, enhancement or some cooperativity or something should be addressed in future. Thank you.
Thank you. So any other questions? If no, I would like to close the session. So please join the speakers.